Thank you. It's great to be here and to share with you a bit of our work in the clinical classification of variation for disease causality. I did put my slides on a Google site that you can publicly download them. The QR code is that little code right there. I'll also have it at the end of the slide deck if for some reason you miss it. But in case you actually want to download and follow along, you're welcome to download the slides. So I'm going to get started. I'm going to talk a little bit about ClinGen because a lot of the work that we're doing in terms of variant classification centers around efforts within the clinical genome resource. And so this program's now been around for about 10 years. The goal is really to create authoritative centralized resources for both gene and variant curation and classification. And we sort of work to aggregate genomic data and then curate on 4 different domains. So gene disease validity, is this gene really implicated in this disease? Variant pathogenicity, dosage sensitivity, which is really around if you add or delete a full copy of a gene, what's the effect on phenotype, particularly coming from cytogenomic microarray testing is where this question comes up a lot. And then finally, clinical actionability. So if I have a pathogenic variant, is there something you could do to a patient that would improve the outcomes by having that information? So we curate on all those domains. Most of this work is led by 3 core U24 genomic resource grants through NHGRI, a part of the Human Genome Research Institute in the US. And then there's now a recent somatic cancer grant that Obi and Malachi Griffith have. And then there's an increasing number of smaller disease-focused grants from different NIH institutes that are being funded. And that's how a lot of the ClinGen program works. The leadership is here. We have a lot of staff across those different grants that are funded, but we actually make use of over 2,800 volunteers around the world that are on over 123 expert panels doing gene and variant curation in different disease areas. So if we think about the curation ecosystem, I sort of think about it as the community is sharing data in terms of variant classification in ClinVar. And I'll talk a little bit about GenCC, which you may or may not have heard of that database. It's a newer database we've launched called the Gene Curation Coalition. And that's where groups are now sharing their interpretation of gene disease relationships. But then on the right side is where ClinGen expert panels are curating through expert consensus information that's then shared from different groups in either ClinVar or ClinGen for variants or genes. And even though the main focus and interest of this group I know is in variant classification and how we think about the effect of variants, you really can't classify a variant until you understand the role that that gene has in disease or not. And so we generally start with the question of gene disease validity. Before I classify a variant, I can't call it pathogenic if I haven't established that gene's role in disease. So we developed a framework in ClinGen, a semi-quantitative framework a number of years ago that would assess the evidence across different types of evidence, both genetic evidence, case observations, de novo occurrence, segregation, as well as experimental evidence, animal models, expression, rescue, things like that, and then aggregate that evidence together to classify each gene disease relationship in one of these tiers: definitive, strong, moderate, limited, animal model only, no known disease relationship in humans, or even disputing or refuting that claim. And then there's additional professional guidance from ISMB that then guides how those tiers are used in clinical genetic testing, with the basic premise being, if you're developing a disease-targeted multi-gene panel, only use genes that are moderate and above and exclude the limited and below. Now, when we do exome and genome sequencing, we look at everything anyway, so those genes are still in there, but then those classifications help contextualize a variant if you report it in a genetic test report, for example. So we score all that evidence. You can sort of see the point ranges, and there's a whole very long SOP that our curators use to assess all this evidence, capture it in a data system, and then result in the final classification. When there's conflicting data, often the experts need to sort of evaluate the validity of those conflicts to decide what ultimately to conclude. But then that final conclusion is then put on the gene-disease relationship. It goes on our website, and then also gets submitted to GenCC. And so to date, we've classified 2,729 gene-disease relationships across many different clinical domains. And keep in mind that any given gene can actually be implicated in multiple distinct diseases. So it's not just 2,729 genes. In fact, the unique number of genes is up there at the top, 2,234 genes under those 2,729 gene disease curations. But what you can see in the summary up on the left, colors are very different than on the screen, but that's okay, is that about 23% of these curated gene disease relationships have insufficient evidence. And mind you, all of these are published claims. These are not just digging up things unpublished. So it just really goes to show that just because a publication is peer reviewed doesn't mean the data there is valid in terms of the claim. And the way that labs and others are implementing this, I told you there was that professional guideline from ISMB. So as an example, in Vitae, as soon as these curations come out, they will change their gene panels. And what you see there is the Brugada syndrome panel that they used to have 20 genes. The experts curated and disputed 19 of the 20 genes that were claimed to be involved in Brugada. They immediately changed their panel to be 1 gene instead of 20. And then there's this process that you can actually add on preliminary evidence genes, but they're not there by default. Similarly, the ASCO guidelines just recently came out guiding labs as to what genes to put in panels, and those very heavily draw from the ClinGen guidelines and the curation of the genes that have been done by ClinGen to guide what genes go into tumor and cancer panels. So I mentioned this GenCC database. We actually started this coalition a number of years ago, trying to get harmonization between how databases like ClinGen, OMIM, Orphanet, the Genomics England panel app, which was public at that time, were defining gene disease relationships because they all had different systems. Some used color schemes. Sometimes the color schemes were opposite in different databases. It was a mess. So we went through a Delphi survey process, got everybody to align on the same evidence-based approach and terminology system. It actually largely mimics what ClinGen had come up with, but we added one additional category of animal model only that we thought was useful. So that became the final set, and then everyone's data was mapped to that. And we also, in the interim, discovered there were a lot of clinical labs curating gene disease relationships in order to build their genetic testing panels, and they were willing to share all that data publicly. And so we brought all this data together. It's now in the GenCC database. I like to call it the ClinVar for genes, because I mentioned earlier this is really claims on gene disease relationships. And to date, we have 22,060 claims on 5,165 genes from 12 different submitters. So it's a fairly large data set. It's tenfold larger than what ClinGen has done to date. So it's a way to kind of crowdsource the problem until ClinGen is able to get every gene disease relationship curated, which is our goal, but it does take time when you're doing this by a large group consensus process. So here's just a screenshot of the interface. I've typed in GJB3, but you can type in your favorite gene, see if it's been curated and by whom. And then what we also can do, because all this data is harmonized, is that we can look for discrepancies. And then we have monthly calls across all the submitters, and then we work to resolve any of those discrepancies. And we sometimes push them to the expert panels to work out. So that's a little bit about the GenCC database. This is the example I alluded to from the Brugada syndrome, where a lot of those genes were disputed by the experts for that particular disease. And this is another example from our hypertrophic cardiomyopathy group. And one thing you might notice, so they've stratified the evidence by genetic in red and experimental in blue. And what you might notice in the area of limited evidence, that a lot of these genes only have the blue evidence and very little red. What I've noticed over the years is a lot of the false claims end up relying really heavily on functional assays that don't always recapitulate human physiology and disease. And I think that's where some of these can go awry when they're claiming. They say, oh, I saw a missense variant in a patient, so that was my candidate, and then I went and validated a functional system, and that system really didn't really truly mimic the human disease phenotype. So that's just an example. But now we're going to move on to variant pathogenicity, which I know is the major interest here. So I talked about ClinGen's role and the ClinGen expert curation. However, experts working at like a few variants a month are never going to classify the vast majority of human variation. We have billions of variants in our genomes. So there's no ClinGen expert panel is going to be formed for every gene and classify every variant. I guarantee it. So we have to think about different strategies in our community for how we interpret and annotate and understand human variation. And the way I've thought about it is to sort of take a four-tiered approach. So one is to develop common standards for how we look at evidence and classify variants. And if we can put those standards and disseminate them and teach people how to use them, then the likelihood that every individual person or group classifying variants is more likely to do it similarly will increase if we have standards, right? So that's step one, is to develop the standards. I'm going to talk a bit about the ISMB effort underway right now on the next version. But in addition to putting out common standards, publicly sharing your own variant classifications is critical. So we've largely centered around submission to ClinVar as the primary way that whether it's a clinical lab or a research group shares their variant classifications. This creates transparency. It crowdsources the work. They're not always going to be the same. Why? Because there is subjectivity to how you interpret evidence. Also, many labs are sitting on different underlying data that's not actually publicly accessible. These are patients they've tested, observations they've made, segregation studies, de novo occurrence. These are all data types that are often observed in a clinical lab and never published on. And sometimes when the lab shares their evidence for why they classified, you'll see, we observed this variant as a de novo variant in a patient with disease X. Things like that will be in those evidence summaries. Not always, but many of the labs are sharing that. Or at a minimum, you at least know they saw it and you can reach out to the lab and ask. And a lot of times those labs will then divulge the detailed evidence. And in fact, a lot of this data comes out in the ClinGen expert panels because we involve the members of many labs and research groups, and they bring their data to the expert panel, and we can get access to that data. I wish there was a more easier way to get access to that data, but nonetheless, it's much better with ClinVar and sharing the interpretations and figuring out where that data exists than where we would be without that. And then the next is really accessing a shared evidence base, and I'll talk about some of the strategies for that in a second, but that in the end is really going to make a difference, is how we really work to start sharing the underlying evidence. So I'll talk a little bit about that. And then lastly, as I mentioned, engaging experts in this systematic consensus-driven curation. But as I mentioned, it's only going to be a small fraction of variants that we can do that for, so we have to pick out the ones most worthwhile. And so those are the conflicting in ClinVar where groups disagree with each other, or sometimes they're everybody agrees it's a variant of uncertain significance. But if you get the data from all those groups together, you can sometimes push it up to pathogenic or down to benign by bringing that data together. And those are the areas that the expert panels focus on, given that they can't curate everything. So I mentioned that first step is really the putting out common standards. So I'm going to spend a little bit of time on where we are right now and hopefully where we will be sooner than later with the V4 version. So many of you who work in this field are probably familiar with the ISMB AMP standards for variant classification that we published in 2015. You might remember the rainbow chart, which doesn't look very rainbow from this view, but it is. And that's where we developed common terminology and a basic framework for looking at evidence and evaluating and understanding the strength of that evidence. We had then immediately formed a sequence variant interpretation working group within ClinGen. with the goal to really refine and evolve those guidelines as they were tested and deployed by the community. Also, we manage all these expert panels I mentioned, and so this group is reviewing and harmonizing the gene and disease specifications for each of those different disease areas. So what's the allele frequency that is the threshold to call a variant benign for this gene? Or how good is this functional assay for this gene? Things like that that are specified by the expert panels, and we review those submitted specifications and make sure they're not going off the rails in terms of how they're approaching that. And then generally with the goal of moving towards a more quantitative framework than where we were in 2015. So that work has been ongoing. And if you go to the ClinGen SVI website, you will see lots of different links. Some are published papers, others are just white papers, but they're really giving a lot more detail to how to evaluate and quantify these different evidence types that go a stage beyond what the 2015 guidelines did. And most all of this work is being incorporated into the current V4 guidelines that we're working on right now. We also have a criteria specification registry. So all the individual expert panels that develop their specifications, they all then get deposited in the C-spec registry. Of course, all these URLs are in my slides that you now have a QR code or will at the end, so you can get access to all this stuff. We also, in the last year, came out with the guidance for low penetrance risk allele, which are a challenging set of variants. I won't go into too much depth, but happy to answer questions about that. But really trying to distinguish the really low penetrant that are really shouldn't be using the term pathogenic and instead use risk allele, kind of more on the GWAS realm, versus what appears to be a variance in a monogenic gene, but with significantly reduced penetrance, where we call those pathogenic, low penetrance, and then everything else. So trying to distinguish and then provide some analytical framework to evaluate the validity of these risk alleles. So onward to now the current variant classification guidelines. So these weren't numbered and versioned, but we are retrospectively numbering them in this next V4. And so the one that will come out that's under development right now will be V4.0. We then retrospectively labeled V3, V2, and V1 in going backwards. We also are going to label it 4.0 with the goal that we will be a standing committee as opposed to just convene for a guideline and then break out, which is what we did before, and it's happened every time. So we'll keep going with this committee, and then we'll try to come out with updates instead of like throwing it out there and then letting ClinGen develop the additional guidance. We're now, ClinGen is a part of this guideline, and we as a collective group will come out with iterative updates of the ACMG AMP CAP ClinGen guidelines. We're also have been working back and forth to harmonize across CNVs and sequence variant classification, and they've now reconvened the CNV committee to use the Bayesian framework as well. So that's going to be gradually harmonized. At some point, it'll be a single guideline that covers everything, but we're not quite there yet. So this is the working group. We've been working for like 3 years now, I think it's been. We just did our third in-person 2-day meeting last week, I guess it was. So we're getting close to having a draft framework developed that can then start to be piloted. Some of the limitations of the 2015 guideline were issues around subjectivity in when you apply evidence criteria, ambiguity around the strength of certain criteria, lack of clarity around combining criteria and when you're actually double counting evidence that kind of falls into two different evidence buckets. Also, lack of guidance of how to combine a criteria when some of the evidence is towards pathogenicity and some towards benignity. And so how do you merge that together when the guideline didn't really clarify that? And then recognition that certain criteria were just totally inappropriate or misused. One of those being PM2 absence in the population. And we've come to really appreciate that absence means really there's not evidence. You need a population frequency that's consistent with pathogenicity, but simply being absent is not in itself evidence. We all have unique variants in our genomes, and just because they're unique to us does not give you evidence for pathogenicity. So that whole criteria will be gone in the next guideline. Now, the reason we haven't taken it away yet through ClinGen is because we never let any of the other evidence types get strong enough to make up for getting rid of PM2. And that was a fundamental challenge in sort of evolving this in pieces. So in the new guideline, other really good evidence will allow higher scoring, and then you won't need this crutch of PM2 to help get you to pathogenicity. And then there was sort of the PP5, BP6 reputable source that was specifically put in because of access to Myriad's classifications without their evidence, but it's been horribly misused by people just saying, it's called X in ClinVar, therefore, so that's going away. So in the new framework, it'll all be based on the Bayesian framework that Sean Teptijan published in combination with ClinGen's SVI group. And that was really looking at all these strengths of evidence and mapping them to a Bayesian framework, which was then adapted to a point value system. And so you can see the odds of pathogenicity in one column, then mapped to the point value to make it a little easier on the math side for our community. And then that then maps to the ranges you see at the top, where 0 to 5 points is VUS, 6 to 9 is likely path, 10 or above is path, and then so on the likely benign side with minus values. So that's what our framework will be based upon in the next version. This is a little harder to read slide, but just to give you sort of a draft version, and I can tell you already some of these codes we just changed, so don't rely on the 3-letter codes that are in here because a bunch of them are changing. But if you can see into here, it sort of separates human observational data, which then breaks into counting cases versus locus specificity. And interestingly, we just published a paper within the last, 6 months around segregation and phenotype specificity, arguing that they are inherently doing the same thing, which is focusing in on a specific locus. And you can do that either through linkage, through the segregation of variant with disease or gene with disease, or the more specific a phenotype is, it will narrow you down to a specific gene. But those are inherently linked. And if you had 100% phenotype specificity, you can't actually add more to it through linkage. And vice versa. If linkage gets you 100% to a single gene, phenotype can't add any more to it. So you're sort of, but when you have non-100% from one or the other, you can add them together and get to a same level. So that's all been described and published recently, and then we'll use that framework in the new guidance. The bottom half is around functional and predictive data. Again, these things are also inherently linked. If you had the perfect functional assay that absolutely recapitulated human disease, and you could say with 100% certainty the output of that functional assay was X, Y, or Z and trust it, then predictive data adds nothing to that, right? On the other hand, if you had a predictor that was 100% accurate, which none of them are, but if you did, then you don't need functional data, you will get to 100%. Where we are with most genes and variants is that the two are not, either is not 100%, and you are having to combine them, but there is some overlap. They're essentially, one's predicting and one's showing you experimental data. Neither is perfect. Combining, they add value to each other, but there is some overlap. And so we're sort of coming up with frameworks for how to add in ways that aren't 100% additive. And we cap at certain levels, and it's a little bit of judgment going on there, but we're working on sort of that framework. So that gives you a sense, and then each area has a set of points, and there's decision trees to arrive at, and this is just a screenshot of the missense variant decision tree. And at a high view, I've circled or squared, whatever, the top, which is the use of assessing the impact of the missense change, the amino acid substitution. But what everyone now has to do for every missense variant is also assess its splice impact. And so there's actually 2 decision trees you go down. And at the very end, you look at what point value is higher, and then you use that. And so that will be a sort of framework for every missense variant that has to go through that. So if we zoom in at the top of that to the missense part of it, it's starting with in silico predictors, and the group that worked within ClinGen SVI to validate these predictors have given assigned point values based on how well they correlate with pathogenicity from validation. And they're actually in the process of validating some other tools. I know alpha missense is in there. I forget which other ones, but you'll be able to look up your variant in one of these tools and get a score, use those points. We also have a transcript assessment. We were really finding people weren't paying attention to the fact that certain exons are missing from other transcripts, and putting a variant in that exon has no effect because other valid transcripts don't have that exon. So we put transcript evaluation of every decision tree. And then we usually have a section that is comparator variants. So I have another variant at the same amino acid position, or another splice site variant in the same splice site, and with different point values depending on how similar that variant is. So these are commonalities around all these decision trees and giving guidance to point values for different types of evidence. And then, as I mentioned, at the very end, you'll get a score for the missense assessment. I haven't gone through the splice, but you'll get a score for that. And then you look at which is more positive, and you use that, because the variant usually is going to operate through one or the other. Occasionally, it could actually operate through both, but we're trying not to get distracted by rare scenarios to try to get the basic framework out. So you'll take the more positive value and then score that. And then there's also, and this is the least well-developed, so you don't want to zoom in too closely on this figure, but we're refreshing some of the functional assays and how we score them, and how many controls need to be there for benign and functional, and giving point values based on the extent of validation, as well as the odds ratio in terms of that data compared to controls and the validation of it, and then giving guidance on how to score that. And then, as I mentioned earlier, how to then combine that with the predictive data to come out with a final point value. So I'm just going to give kind of an example of, so the committee is actually testing out our decision trees right now. So I tested out some variants through our hearing loss curation panel. And this is an example of a oocyte coupling assay that was published in like 6 different papers and had controls in those. And so there were a total of 23 path and likely path variants used to validate, and 4 likely benign and benign. And that led in our original framework to a proposed strength of PS3 moderate. Now that in the new guideline will correlate to a point value, but just to give you a sense of this type of data. And so interestingly, in the original, using the 2015 guidelines specified for the hearing loss group, that in vitro functional data was scored as moderate, but the case observations gave very little evidence. PM3 was the level. In the new framework, so you'll see pop freq, that's the absence from nomad, population frequency, always scores either 0 or minus. You never get a plus value. That goes back to my comment about PM2 is gone. So in this case, the variant allele frequency, it was absent from nomad, so it gets no minus points. It gets a 0. But then the case observations, there were 3 cases with likely path variants. This is a recessive gene, but they weren't phased, and so we give points for when they're known to be in trans versus not known, and there's different point values per observation, so there's a lot of detail to how to score every case. Also, usually the expert panels give scoring based on the strength of the phenotype. Is this a very specific phenotype or not? So those case observations get scored, and that was 4.5 points for those 3 cases with in trans but not phased variants, or I should say compound hets but not phased. And then the Revel score was not high enough to score any points, and spliced AI had no predictive impact for splicing. But then that functional assay got 2 points. So together that got 6.5 points and scored likely pathogenic. On the old framework, it only got to a VUS. I actually think this variant is likely pathogenic. And in fact, the ClinVar submissions from labs, even though formally it didn't score with the framework, most of them were calling this variant. 3 out of 4 labs called it likely path. One that was much older submission called a VUS. So I actually think the new framework is doing a better job of scoring this data than the old framework. I know this is one example, but I've been doing a few of these. So that just gives you some flavor of what's to come. We're hoping that in the next month or so that this will be ready for the first round of testing. We have sort of 3 phases of gradually larger and larger sets of groups. We have 90 groups signed up to pilot these variants. So that's coming soon. I also, let me just skip ahead to this slide. So I noted that the VUS category is 0 to 5 points. That's actually a really broad category. In the new framework, we're dividing that into 3 subclasses: VUS low, VUS mid, and VUS high. And the point values will directly put you in the subclasses. There won't be any professional judgment required. It's all there based on the point values. So this is really important. And one of the differences that's happening, and this prior slide, my point was 1/3 of all genetic tests end in a VUS and inconclusive. It's a huge problem for our community. Most highest percentage of variants are VUS or conflicting. So we need to solve the VUS problem. Some of that is just allowing our clinical community to focus on things that might be more relevant. So one, it'll be actually easier to get to likely benign in the new framework. And we allow only 1 minus point to get to likely benign. And some people are a little freaked out about that, but the truth is, for wide scale testing where there's a low prior probability, most variants are 90% chance that they're benign, I can tell you. So this will be better matching what's truly happening. If you've got a patient with a highly specific phenotype and a variant in a highly specific gene for that phenotype, you will get phenotype specificity points, and it'll get out at the likely benign. So the only place you should be concerned about a random variant with no evidence is when there's a highly specific match gene to phenotype, and then you score phenotype specificity. So don't be worried. It will be likely benign when it needs to be likely benign. But the use of these subclasses is going to be really critical. And this is just a look at one of the members of the hearing loss expert panel who's taken 179 missense variants, or all VUS, in her lab, scored them on the draft framework. Keep in mind, this is not finalized, so she's using the draft version she had access to, because we've been talking about this, posting on it. And you can see the VUS is now dividing, a lot of them going, 49% of them going into likely benign or benign, 3% becoming likely path, and 6% in this VUS high category. So a lot of variants being able to be sort of taken out of view in a lot of ways by VUS low and below. And that is actually something that's, I think, important for our community, to get the physicians to focus on just those VUS that actually have some evidence. And in fact, some data that we're working on publishing, a bunch of labs, my own lab that I used to direct at Mass General Brigham, along with Quest and LabCorp and Baylor, all have been using sub-tiers, these subclasses, for like 15 years. And one of the things we've found is that the VUS low category have never over 15 years gotten to likely path or path, any of those variants. In contrast, the VUS high group, almost half of those got changed to likely path or path. So these subclasses are going to become really critical for thinking about what physicians should pay attention to and do it, or even researchers do more work on, versus ones to ignore. I'm going to make sure we have time for questions. So I think I'm going to, I have till what time? Until 3 o'clock? Yeah. Okay, then I'm going to stop here, actually, so that we have a little time for questions. You have copies of my slides, which I'm going to just skip to the very end. and leave this up if you want to download any slides, but I'm happy to leave the end for any time for questions. Thank you. Yes. A drag.
Okay, I'll just I am, I guess, a little freaked out about -1 to the 9. It means that the rebel score of 1 to 9 or so will be able to completely classify it, and I think that might be a problem for the genes that rebel hasn't seen in training, but it will be probably just fine for the other genes. And I had another question, but I will ask that later.
Yeah, I mean, if anybody can come up with data to suggest that it is the likelihood of any of those variants getting to pathogenicity is higher than 10%, which is what it has to be be beyond likely benign, then I'd love to see that data, because I'm not convinced that the likelihood of pathogenicity is above 10%. But, I think any evidence that you have can get it out of that -1. As I mentioned, if the gene that variant is in is a good match for a phenotype, you can score phenotype specificity points, even in the absence of actual variant level evidence, just the phenotype to gene match. So there, if that is true, and the likelihood of pathogenicity is, higher than the 10% range, but there's other evidence. I think the question, and since you're part of our sequence, our in silico predictor group, like, understanding how to score these predictors, if we're not doing it perfectly now, then let's figure out how to evolve them to get there. But I still feel pretty confident that it's not 0% chance of pathogenicity. Is it above 10%, right? So you really think?
Overall, it may not be, but there might be a large group of genes where it will be that cannot.
Yeah, there. Yeah, and I think the point about gene specificity around these predictors, there's definitely these predictors can work better or worse for certain genes. And this is where we will rely on our expert panels to define the predictors that work best for a particular gene. And that could help, kind of deal with some genes for which the predictor today that's largely being used, a lot of groups use Rebel, is not optimized for that gene, and maybe there's a better predictor. Or you could say, works well for this region of the gene, but not the whole gene, and define those regions. So I think there are ways that we can help improve this over time, but I still think that the biggest problem right now is patient harm being done because of the VUSs that are being act as if they're like, oh, they must actually be pathogenic. The lab is just not willing to say that. And that's causing equal harm to potentially a rare variant here or there getting misclassified as likely benign. So it's a balance. Yes, question?
Yes, thanks a lot. So just how much do you believe epistasis is impeding the predictability for pathogenicity? Or the other way around, how much doesn't it matter?
So, I assume what you're getting at is, to what extent could other variation in a gene impact the pathogenicity expression of a variant, right?
Okay.
So a variant on the background of this haplotype versus a variant on a background of a different haplotype. And that's absolutely a huge challenge today. And I think we're not going to get at the answer to that question until we have better genomic data being shared. Because right now you go into ClinVar and you have a bunch of patients with that variant, but you have no idea what their haplotype is, right? And I very strongly believe that the penetrance and expressivity of variation is going to be heavily influenced by other variation, both in the same gene that's insists in that gene, and probably in other genes or their products in the same pathways. But until we have a lot of data sharing and ability to look at the haplotype these variants are on, it's hard to answer that question well, right? So I think it's got to be something we need to focus on in the future. It's just, going to be a little bit till we really get good data sets shared that you can access that data, right? Yep. Thanks.
Yeah.
I assume I'm out of time.
I guess one more question. Yeah.
One more?
So indeed, you're in front of people that develop new software. So what do you advise to work on? What's the most pressing problem?
Advice in terms of developing a silico prediction software, is that the
Yeah,
question?
what type of variants or processes we are poor with current predictors?
Yeah, I mean, so I have been quite impressed by Splice AI and really the difference in accuracy. Like, we've been validating a lot of Splice AI predictions with RNA-seq data and turning out to really pull things that we never even would have paid attention to, cryptic splice sites and things. And that was really developed through a machine learning approach, neural network, really trained off the whole genome data set. So I think, ability to really train these predictors on a lot of good data, I think will help them, and not just pathogenicity in ClinVar, but other data that is functional data and large data sets with actual phenotypes and genotypes that we can draw from, even without somebody trying to classify the variant, which has its inherent sort of biases that then bias the training. There's this whole sort of bias that goes into it. So the more we can use really raw genomic and phenotypic data to help train, I think we'll build better predictors than we have today. That's one of the reasons I spend so much of my time in genomic data sharing, is because that's what we need to improve the work that we're all doing.
Thank you.